Every time you learn something new, a name, a fact, a skill, you are not just adding information to a mental hard drive. You are physically and genetically altering the cells in your brain. This isn't a metaphor. A signal, born from experience, travels through a neuron and arrives at the nucleus, the cell's command center. There, it reaches past the nuclear membrane and interacts directly with your DNA. Specific genes that have been dormant for hours, or perhaps their entire existence, are suddenly awakened. They are transcribed into RNA, which then serves as a blueprint for new proteins. This process, called activity-dependent gene expression, means that your experiences are actively rewriting the operating instructions of your own neurons. To understand how this happens, we have to look at the space between neurons, the synapse. Think of it as a microscopic gap where a conversation happens. When one neuron fires, it releases chemical messengers called neurotransmitters. These chemicals cross the gap and latch onto receptors on the next neuron, creating a small electrical pulse. If this happens repeatedly, the connection gets temporarily stronger. This is called short-term potentiation. It's like a quick, firm handshake. But this handshake is fleeting. The memory of it, the strengthened connection, will fade in minutes or hours unless something more permanent occurs. The conversation has to become a commitment. For a memory to last a lifetime, that synaptic signal needs to do more than just excite its neighbor. It has to send a messenger to the one place that holds the power to enact permanent change, the nucleus. This is where the electrical becomes biological, where the ephemeral becomes structural. A cascade of chemical reactions triggered at the synapse travels inward, down the neuron's internal scaffolding. Its destination is the very heart of the cell, carrying a simple but profound request. This connection is important. Make it permanent. This is the trigger for late-phase long-term potentiation, the molecular basis for long-term memory. Once the signal arrives, the nucleus doesn't activate just any gene. It triggers a special class of genes called immediate early genes, or IEGs. Think of them as the cell's genetic first responders. Genes like C-Fos and ARC are primed for rapid activation. They don't need any new proteins to be made to get started. They are ready to go the moment the call comes in. Within minutes of strong synaptic activity, these IEGs are being transcribed into messenger RNA at a furious pace. They represent the cell's acknowledgement that something significant has happened, something worth remembering. And they are the managers who will organize the permanent construction project. But the proteins made from these immediate early genes are not, for the most part, the building materials themselves. They are something even more powerful, transcription factors. Their job is to turn on a second, slower wave of late response genes. These are the genes that code for the actual bricks and mortar of memory. They produce robust structural proteins, new receptors for the synapse, and molecules that act like glue, solidifying the connection. These newly manufactured components are then packaged and shipped all the way back to the specific synapse that made the initial request, a journey across the microscopic landscape of the cell. But there's another layer to this. Your DNA is not just a loose string of code. It's tightly wound around proteins called histones, like thread on a spool. For a gene to be read, the spool has to be loosened. This is where epigenetics comes in. Learning doesn't just send a signal to read a gene, it sends a signal to make that gene readable in the first place. Chemical tags, through a process like histone acetylation, attach to the histone tails, forcing them to relax their grip on the DNA. This exposes the memory-related genes, making them accessible to the cell's machinery. Learning, at its core, is the act of physically unwrapping your genetic code. This entire biological saga, from synapse to nucleus and back again, is what turns an experience into a memory. It physically alters the structure of your brain. But here is the truly strange part. The process isn't a one-time event. Every time you recall a memory, it seems the memory becomes unstable again for a short period. It has to be resaved, a process called reconsolidation, which often involves the very same gene expression mechanisms. This means a memory is not a file you retrieve, but a structure you rebuild each time you access it. And during that rebuilding, it can be subtly changed, updated with new information, or even distorted. Your memories aren't etched in stone. They are written in a biological ink that is constantly, quietly being refreshed.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.